All right, the last thing I want to talk about in this Unit 2 introduction is membrane permeability. And that's because this is one of the most important aspects of the membrane in regulating what gets into the cell and what gets out of the cell. And that regulation is really based on the permeability through the membrane itself. And then some things are going to need help via channels or transporters, and other things get through pretty easily on their own. So what I want you to do is think about the properties of the phospholipid bilayer and the cell membrane that we've talked about already, and then see if you can figure out which of these various molecules here, we have oxygen, triglyceride, carbon dioxide, etc., which of those are able to get through the membrane on their own without help, so without needing a channel or transporter, which ones permeate through the membrane on their own rapidly and easily, and which ones have more difficulty, basically would need help. So put those in order from the ones that get through the membrane the easiest to the ones that get through the membrane the hardest. And pause the video for a second and just really think about it and try to put those in order. All right, so let's talk about this for a second. There are two aspects here that really regulate this. There are two aspects. Those two are polarity and size. And I'll say this right now, for the most part, polarity trumps size. So if you're going to get through the membrane, let's look next slide here. Right, we know this membrane is hydrophobic in the interior. And so if you are polar, you're going to hit that hydrophobic interior and basically be repelled. And if you have a charge, you're really going to be repelled. Basically, charge is like superpolarity. And so anything with a charge is repelled by that hydrophobic interior quite rapidly. But anything that's hydrophobic is going to make it through pretty easily because it's not repelled at all by that hydrophobic interior. And you might actually ask why it's in the polar part in the first place, but these things are so small that they actually dissolve in the aqueous solution, but they make it through this hydrophobic part right away. So non-polar is going to get through much more easily than anything with polar, and polar gets through more easily than anything with a charge. Now, the other factor is size. Anything smaller is going to be able to make its way through this membrane much easier than anything larger, right? So if you have a big thing of Jell-O, for example, and you throw something small at it, it's going to penetrate easier than if you throw something huge at it. So huge is not going to get through quite as easily. But for the most part, polarity is more important. So here's the way I like to think of it. Anything small, non-polar, is going to make it through really easily. Then you kind of have larger non-polar. So if it's huge non-polar, then it still might not get through real easily, but a large non-polar one is going to get through more easily usually than anything that's a small polar. So you have small non-polar, then larger non-polar, then you have small polar, then larger polar, and then you have small charge. By that time you're really not getting through the membrane at all, and anything larger charged won't get through. So as we continue to think about this, essentially we have our small hydrophobic molecules that get through the easiest. Then we have larger nonpolar molecules, followed very closely by some of those small uncharged polar molecules. Now, both of this set, depending on the size and the polarity, they will probably get through on their own. We know water can get through the membrane, but we also know water gets through the membrane relatively slowly. So these can get through, but they might be slowed down a little bit, whereas oxygen, carbon dioxide just make it right through without any problem at all. And that's actually really important because if oxygen and carbon dioxide needed help to get through our cell membrane, it would be a lot harder for our cells to obtain the oxygen and get rid of the carbon dioxide that they need for cell respiration. And then we get to the parts that really don't get through. So if you have a large polar molecule and basically anything that has a charge, then those things are not going to get through the membrane without help. So again, in quick summary, first is small nonpolar, then you have larger nonpolar, then you have small uncharged, then you have larger polar uncharged, and then you have ions, anything with a charge, and obviously things like DNA, RNA, and those larger charged molecules like proteins, they don't get through at all because not only do they have a charge, they're also huge. All right, so with that in mind, the last thing I want to do now is just kind of give you a little bit of an example of something that we're going to tie through this entire course. So I'm going to talk a little bit about caffeine and why caffeine works the way it does. And we'll revisit this a few times because this has several concepts that are going to be taught in several of our units. So the first concept with this actually involves membranes, and that's why we're talking about it now. So calcium is highly regulated, and for the most part you have really high levels of calcium in the ER, or if it's muscle, in the sarcoplasmic reticulum, and you have relatively low levels of calcium in the cytosol, and then higher levels of calcium extracellularly. So in order to do that, calcium has to be pumped from low in the cytosol to high in the ER, or from low in the cytosol to high in the extracellular space. And then when a channel opens, the calcium is going to flow really rapidly from high to low, from the ER to the cytosol, or from the extracellular matrix to the cytosol. And this is important because this means that you can regulate a lot of things in the cytosol by whether or not calcium is there. And one of the things we'll learn about in this unit is regulating the release of neurotransmitters by opening calcium channels and allowing calcium to flood in. But another thing that's regulated by calcium in the cytosol is muscle contraction. And this is true for the heart and other muscles. So muscles have the sarcoplasmic reticulum. This is kind of like the ER where the calcium is being stored. And then when a neuron stimulates the muscle to contract, that calcium will be released because calcium channels open. And then that calcium is really important because they're going to bind to this molecule called troponin. And we'll learn all about this in Unit 5. But troponin, basically, when calcium is bound to it, gets out of the way, and that muscle now can contract. Right? So when calcium is in the cytosol, it's going to allow contraction. And then for relaxation, you have to pump the calcium back out, and that's done through this pump called phospholambin. And so you think about it, when your heart's beating, this has to happen quite rapidly. So when your heart contracts, calcium is released. Then when your heart relaxes, calcium is pumped back, and it keeps doing that with every beat. Boom, 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 boom. So that calcium movement is actually quite rapidly, although we all know that it's much more rapid through a channel than through a pump. But this is very important that this happens over and over again. Right? So calcium's released, binds to troponin, muscle contracts. Calcium's pumped back out, muscle relaxes, and this basically cycles over and over again. All right, so let's think about what that mocha latte you had this morning to try and stay awake during watching this video. Hopefully it's working. What is that going to do? So in order to understand that, we first have to start with adrenaline. So epinephrine, or adrenaline, causes activation of a signaling molecule called adenylyl cyclase. And we're going to talk a little bit more about this in Unit 4. But that signaling molecule then leads to cyclic AMP production, and cyclic AMP leads to protein kinase A activation. And you by now know what a kinase does. What does a kinase do? Well, a kinase phosphorylates. So pKa is going to phosphorylate troponin, and pKa phosphorylates phospholambin. By phosphorylating troponin, the affinity for calcium is higher. So troponin is going to bind to a calcium much faster. And so that leads to faster contraction. And then phosphorylation of phospholambin actually makes that pump work faster. So not only do you get faster contraction, you get faster relaxation. And that makes sense because if you have an adrenaline rush, your heart starts beating faster because you're getting ready for the fight or flight response. So phosphorylation at the end of the signaling cascade causes a functional change in troponin, a functional change in this pump, and your heart beats faster. All right, so how does that relate to caffeine? Well, we just talked about this signaling cascade: epinephrine, adenylate cyclase, cyclic AMP. Cyclic AMP leads to pKa activation. pKa phosphorylates troponin, right? But we also don't want caffeine to the adrenaline effect to last forever. So if troponin is becoming phosphorylated, what do you think is naturally at work to dephosphorylate it? Well, there's going to be a phosphatase, and phosphatase is constantly removing the phosphate groups. And we also have over here something to turn this signaling cascade off. Cyclic AMP is constantly being turned into AMP, and no more cyclic AMP, you have no more pKa activation. So if you still have adrenaline, then you're going to still be making more cyclic AMP faster than it's broken down. Kinase is still going to be active, and you're going to phosphorylate troponin faster than it's dephosphorylated. Well, let's think about it. Once your adrenaline rush is gone, there's no more adenylate cyclase activated. Therefore, cyclic AMP is no longer being made. Phosphodiesterase continues to work, and your AMP levels rise, your cyclic AMP levels lower. And if your cyclic AMP levels lower, pKa is no longer active, and so now you're no longer rephosphorylating troponin, so the phosphatase is still working, and everything will eventually become back to the dephosphorylated troponin. Your heart rate relaxes a little bit more, the adrenaline effect goes away. Alright, so what caffeine does though is caffeine actually inhibits phosphodiesterase. So if you inhibit phosphodiesterase, the cyclic AMP levels are no longer being made to AMP. And so let's think about this for a second. Now, even if your adrenaline levels have decreased, you're no longer reducing the cyclic AMP. The cyclic AMP remains high from that time you had that adrenaline rush, and so the kinase is still active, and you're going to continue to hold on to that much more rapid heartbeat. Basically, you maintain the adrenaline rush for a much longer time because caffeine inhibits phosphodiesterase. So when you wake up, that's not a huge adrenaline rush, but it's more than you had when you were asleep, and so caffeine kind of maintains that state of awakeness. BAM! So what I just did is I just made that mocha latte work for you, because really, in order for caffeine to have any effect, you have to have an adrenaline rush, and then caffeine is going to prevent that adrenaline rush from going away. So you're welcome. All right. So we're going to keep revisiting this. This has a lot to do with Unit 4 and signaling cascades and the fact that the off is always on. We're going to talk about this with regards to neuronal signaling and membrane like calcium in this unit, but we will also talk about it in several others as well, including muscle contraction details at the cellular level in Unit 5 and so forth. So anyway, hopefully that was interesting to you, and I look forward to seeing you together on Friday. Thanks.